So in this chapter, we're going to study what are the details of the components of the bacterial cells, as well with the components of plant cells and also animal cells. So here we have an scanning electron micrograph showing you serratia forming a film in this structure. And one of the things that we know before is that these bacteria, in general, they tend to form biofilms. And these biofilms are important for the survival of these microorganisms and the colonization of several places, including catheters. So the first objective that we're going to consider is what is the cell structure of prokaryotes and eukaryotes. So if you remember, prokaryotes are organisms that they don't have a DNA enclosed by a nuclear membrane, while eukaryotic organisms, they do have a DNA that is enclosed within a nuclear membrane. So here is an overview of the general differences. So prokaryotes, they have one circular DNA, chromosome. With few exceptions, they only have one chromosome. Sometimes you can have organisms that have more. And then these prokaryotes, they don't wrap around the DNA in histones like eukaryotic cells do. And there is no organelles enclosed by a phospholipid membrane in the prokaryotes, so meaning like they don't have, for instance, rough endoplasmic reticulum, smooth endoplasmic reticulum. And the bacteria, which is part of the prokaryotes, have a wall made out of peptidoglycan, while the eukaryotic cells, they don't have, the animal cells, this wall, but plant cells do have, and also fungi. Now, for archaea, they have a cell wall, and instead of having peptidoglycan, they have pseudomurine. And one form of division that these prokaryotes have is binary fission. Some might divide by fragmentation or budding, but the majority will be by binary fission. And then eukaryotes, they divide by mitosis. And the cell walls of eukaryotes, they will have a polysaccharide, and they do have organelles, like mitochondria, rough endoplasmic reticulum, smooth Golgi apparatus. And they have multiple chromosomes that are linear. And these chromosomes, this DNA, is enclosed in a plasma membrane. So that's a general overview. So, what is the main characteristic that distinguishes prokaryotes from eukaryotes? But the main one. No nucleus. Mm-hmm. So now let's identify the three basic shapes of bacteria. So, in average, the bacteria measures between 0.2 to 2 micrometers in size, in diameter, by 2 to 8 in length. And most bacteria have only one shape. This is what we call monomorphic bacteria, and few of them will be pleomorphic, have more than one shape. So, if the shape is like a rod, the bacteria will be called bacillus. If it's in a sphere, it's going to be a coccus. If it's a spiral, depending on the thickness and the length, it can be subdivided into vibrio, Aspergillum, and spirochetes. Spirochetes are long and slender. Aspergillum are a little bit shorter than a spirochete and are a little bit thicker, and both of them, they have that helical shape where you have the coils very close to each other, while Vibrio, it's a short coma-shaped bacteria. For instance, Vibrio cholera. And we have resurgence very recently of this disease, Vibrio cholera. And then some bacteria, rarely, but they can have this shape. They are star-shaped, and some of them, they can be rectangular. Mostly these ones can be found in the environment, the last two. So here is a view of these type of shapes. So Vibrio is like a coma, very short, with a little bend. Aspergillum has a spiral-like shape, like spirochetes, which are here in the bottom. So this is the corresponding SEMs. But this one is a little bit more slender. This one is a little bit thicker and shorter. Here we have a star-shaped bacteria. And then here, rectangular shaped bacteria. Now for the arrangement, bacteria can arrange in pairs. In this case, you call it, depending on the shape, as diplococci or diplobacilli. So 2 rods will be diplobacilli, 2 coccus will be diplococci. If it's a sphere that arrange in clusters, like Staphylococcus, you call it Staphylococcus. Staphylo is the Greek for grapes, the Greek word for grapes. And then some of them, they can form a long chain. In this case, you call it a strepto. And then if it's a rod, you would call it a streptobacilli. If it's a sphere, you would call it a streptococci. If they form groups of 4, you call it tetrads. And if they form groups of 8, you call it sarcinae. So here is the arrangement for these different types of cells. So here we have a diplococcus. Here you have a streptococcus chain, tetrad, groups of 4, sarcinae, groups of 8, and then staphylococci. So how does this arrangement form? Well, if you see here in this left side of these 4 panels, you can see the plane of division for the binary fission. So in this case, it's axial, go along the entire cell. So you split a single cell into 2 by binary fission. And that way, if you have a single division, you form a diplococcus. But if you keep dividing these diplococcus as they form and they mature, you form an elongated chain. So that's a streptococci. And then if the plane of division happens in 2 axes, like the Y and the X, you will end up with a tetrad. So a single cell can divide into 4 and then form a tetrad. And then this one, it will be division into axes, X, Y, and Z, and then you can form sarcinae. Staphylococcus is just simply binary fission without organization, so that's why they form a cluster. For bacilli, you have single bacillus. You can have diplobacillus when you have pair. So in this case, this one, it's a diplobacillus because they don't separate. A streptobacilli will be a long chain. And then you can have an intermediate shape between coccus and bacilli. So it's not a perfect sphere. It has a round ending. Like an oval shaped ending. So this is a coccobacillus. So the scientific name can be Bacillus itself when it's written with capital letter, unless you are describing the shape in the sentence, but it's that first word of the sentence. In this case, you have to capitalize, but in general, you can have differentiation between both of them. And of course, the genus Bacillus with this capital letter will be a rod. And how will you tell, for instance, if you have in the beginning of the sentence this word, and then they just let you read that word at the beginning of the sentence, you don't read the rest, and someone asks you, okay, tell me, is this word, does it mean that it's the genus, or does it mean that it is the shape? How would you tell? If both of them, of course, are with capital letter at the beginning.
The genus should be italicized or underlined.
Yes, yeah, by the italics. So here is without italics, but it's supposed to be like that. And this is a light microscopy of Bacillus anthracis. It's a gram-positive bacillus. Something that I can see here right away is not only that it forms these long chains, but these little gaps. So what does these gaps represent inside the cell? How some bacteria make babies?
So like a spores.
Yeah, so those are the endospores. When they are inside, they are called endospores, but once you release them, they will be the spores. So remember, last time we were explaining about the Schiffer-Fulton technique, which helps to stain endospores, but even sometimes you can see them indirectly. Even without this technique, this is gram, so you can still see these endospores. So how you can identify streptococci with a microscope? Yes, if they're in chains. And the shape would have to be? Circular. Okay, now for the structure of prokaryotic cells. So prokaryotic cells have internal components, that are found inside the plasma membrane, and external components that are found outside of the wall. So internally, what you can see in this rod-shaped bacteria, it is a lot of ribosomes. Something that is common to all cells is like they have to have ribosomes, independently of what type of cell it is. In this case, we don't have organelles, but we have ribosomes so that the bacteria can make protein. Another thing that we can see here is the DNA, the main type of DNA, that is not surrounded by a nuclear membrane. And of course, these ribosomes, this DNA, and some inclusions that can be storage of energy, they are embedded within the cytoplasm. So here you have the cytoplasm. And now you have an extra piece of DNA there that is a small circular DNA. This is known as plasmid DNA. And this plasmid DNA, what it has genes that helps bacteria to survive in harsh environments or basically also can help for the bacteria to survive the attack of antibiotics. So you can have antibiotic resistance genes. And then we have the plasma membrane here, which of course, like any plasma membrane, has phospholipids, has channels that allows the entry and exit of molecules. And depending on the bacteria is whether you have just only one plasma membrane or you have two, okay? And then outside of the plasma membrane, this bacteria has this layer of peptidoglycan, which will be the cell wall. And if the bacteria is encapsulated, like in this case this, it will have an outer coat, which is basically the capsule. And it can have extensions like this long pilus or this flagella. So let's look at the external structures of the cell wall. So we're going to see a structure and function of glycocalyx, flagella, axial filament, fimbria, and pili. So glycocalyx, it is a polysaccharide and or a polypeptide. And this is viscous material. It's like jello, like gelatinous. And it is found external to the cell wall. And it helps for the bacteria to adhere to the surface of wherever place it's located. If the glycocalyx is well organized, it's going to be named or characterized as a capsule. But if it's disorganized and loose, it's going to be called slime. So one of the functions of this capsule or this material is not only adherence, but also helps for bacteria to resist attack of the host cell immune system. So it prevents phagocytosis. And this one contributes to virulence. Virulence is the ability of a pathogen to cause a disease. The more virulence of that pathogen has, the more likely that it's going to produce an infection. Examples of microorganisms that has capsule is Bacillus anthracis, Streptococcus pneumoniae, Klebsiella pneumoniae. And then in the case of strep mutans, Vibrio cholera, this extra polymeric substance, that's the name for the material that they have, it helps them to attach to surfaces. Like, for instance, in the case of Vibrio cholera, it helps to attach into the intestinal cells. Now, for strep mutans, which is a bacteria that causes cavities, this glycocalyx that this bacteria has can even use it as a source of energy. When it runs out of glucose, it goes into your mouth, it starts auto-digesting its own capsule, and it starts forming acids out of that, and then it will cause cavities because the acid is going to start eroding the enamel, the calcium salts that makes the enamel. The enamel is the strongest tissue that we have in our body. Imagine how bad are those little acids in our mouth. So here we have an image of Streptococcus pneumoniae. So this is with TEM. So this is the bacteria, and it has like this capsule around. So this one causes pneumococcal pneumonia. For flagella, they are filaments that are adhere on the cell surface. They help for the bacteria to move, and this flagella is made out of a protein that is called flagellin. In the flagella, some bacteria has specific antigens that can be identified, and it helps for the bacteria to be classified as part of one of the members of the species that we, or the genus that we call cerobars. An example is the H antigen from E. coli. We have several H antigens, and then you can number them. E. coli O157H7, it's an example. H7 represents to that specific antigen of the flagella. And then the other one, the O antigen, is found in the cell wall. And there are also several of these antigens. So, flagella has three parts. The one that protrudes is called the filament, that is attached to a wider structure near to the cell wall that is called the hook. And then this one, it is connected to the most internal part that rotates, that is called the basal body. The basal body has rings, pairs of rings that attach to the cell wall and the plasma membrane. And this one helps to make this flagella to rotate in counterclockwise direction or clockwise direction, clockwise direction or counterclockwise direction. So here is the structure of the bacterial flagella. All of this part is the filament. Now, if you magnify this region close to this wall, you will see that the filament, the longest part, it is connected to this wider part that is called the hook. The hook rotates, but not because it rotates alone. It rotates because it is connected to the basal body, which is this, and in this case, the basal body has two pairs of rings. So this is the outer pair, this is the inner pair. And it is basically, these rings are rotating around this shaft, like if it was an engine. So this is a gram-negative bacteria because it has an outer plasma membrane and an inner plasma membrane, which is just called plasma membrane itself. And then one characteristic then of gram-negative bacteria is if they have flagella, that they will have four rings to attach into this region. Now in gram-positives, you only have, as you can see here, two rings instead of four. But you still have that shaft, and all this part is called the basal body, and then you have the hook, and of course you have a filament. And notice how this gram-positive has a thicker wall in comparison to this one before. Let me see if that can play the animation. Yeah, I think so. works. Okay, so not all the bacteria move, so not all the bacteria has flagella. And for those ones that have it, you would classify the presence of that flagella into different kinds. So if the bacteria has flagella all around its cell wall, it will be peritrichous. Peri means around, trichus means hair. So it's a hairy bacteria. Now, if it has it only in one end, and it's only one flagella, it will be monotrichous and polar, because it's found in one end or one pole of the cell. If it has a tuft, like let's say in this case he has what, 6 or so, yeah, 1, 2, 3, 4, 5, 6. So in this case, it's going to be called Lophitrichus or Lophotrichus and polar. And then if it has on both ends, it's going to be polar, but you will call it Amphitrichus. Amphi for both. Can I play it? Let me play this one to see what it says. So those movements that run and tumble, in general, are triggered by the presence of nutrients or the presence of toxins in the environment. And in the case of the movement towards the source of energy, it's beneficial, of course, for the microorganism to follow it. And that movement will be continuous and it will be directed. If it's continuous, the movement, that will be the run. And if it's directed, it will be the tumble. So basically, moving towards stimuli, chemical stimuli, or sometimes even physical stimuli, you call it taxis. Chemotaxis will be specific for chemical stimuli. And then, as I told you before, some flagella with their proteins, like the flagellin, can be subclassified into serobars, like E. coli O157. Serological variant, serobars. I'm not going to play that. So here is an image of a bacteria doing runs and tumbles. So with the run, they can do counterclockwise or clockwise. It depends on the bacteria, depending on how many flagella it will have. And then the opposite direction of rotation, and they can do a tumble. So if it does this, will be a run, and then if it does this, will be a tumble. Sometimes they're like, where? So Proteus, it's kind of an interesting bacteria because it's peritrichous, of course, as you can see there. But the movement that it does with all of these flagella around, it is called swarming motion. And when you put a single Proteus, this bacteria, it's kind of very good to grow in the lab. If you put a single bacteria in the center of a pitcher plate, you will form like dunes. As it's multiplying and moving, it looks pretty nice in the pitcher plates. Now, Archaea will have this motility structure that is made out of archaeolins or glycoproteins that are called archaeolins. These archaea are attached to the cell, and archaea, it's a singular name of this attachment, the plural name for this attachment, while archaeolum means only one single attachment. It's not flagella, but it rotates like a flagella and uses ATP for energy. Some bacteria can have also a structure that helps them to move, and it is found inside the cell. This is called endoflagellum. This is found in spiral-like bacteria, like spirochetes, and it goes from one end to the other, and it looks internally like if it was wrapping around these bacteria in a spiral-like fashion, just like as the shape of the bacteria. And when this endoflagella contracts and relaxes, it will produce motion that is called rotation. So here we have an spirochete, Leptospira. So this Leptospira, this is a scanning electron micrograph, has this yellow structure internally that it is called the endoflagella. It's found here enhancing yellow color. So it almost look like if it was DNA. But of course it's not DNA, it's a filament. And this bacteria has this sheet that helps to keep this endoflagella, of course, inside. This one I would like to see how it moves, the spirochetes. Let's see the movement of the spirochetes. So, Treponema pallidum, of course, is one of these spirochetes, and I told you that this cannot be stained with gram, so you have to use immunofluorescence to stain them. And then when someone is working with these samples, it is advisable that they wear two sets of gloves because they're so slender that even if you have like a tiny cut in your material, it can go into your skin. So, and then this is another view of this axial filament with the cell wall and the outer sheet protecting this axial filament. Now, fimbriae is the plural for fimbria, and this is a hair-like appendage that it is found around the cells that allows them to be able to attach into surfaces. So a bacteria that has fimbriae is able to do or to form biofilms, And an example of a bacteria that has fimbriae is Neisseria gonorrhea, and also E. coli 0157. So here we have presence of fimbriae. So this one doesn't allow to move, but to adhere. Now, some bacteria may have a longer extension, aside from fimbriae, or maybe they even have flagella, but this intermediate size extension is called pili. Pili is plural. You can have one or two. Pilus is singular. And then sometimes these bacteria can use this pili to have a movement that's called gliding or twitching. But the most likely use of this pili is to have conjugation. Basically, a bacteria that has a pilus can conjugate with other. So let's make a little distinction here. So this bacteria that has the pili is called F+. This one that doesn't have it is called F-. And let's say you have your circular DNA there. This one has it as well. Like this is what we call the nucleoid. This is the regular DNA. But this one that has the F+, it has an extra piece of DNA, circular DNA. So this is the plasmid DNA. So this bacteria, the F+, is going to duplicate its plasmid DNA. It's going to make another copy. And now it's going to transfer it into this bacteria that was F-. When this one, that was F-, received this plasmid, now it's going to become F+. Conjugation, guys, is the kind of fancy word for marriage. One day one student said, "What's the species it look like?" And I said, yes, it looks like that. And now the F- that was not aggressive, let's say like it didn't have enzymes to cause a bacterial infection, with that transfer of the plasmid DNA will become aggressive. So virulence factors can be transferred with these pills, with this conjugation. This is basically sexual recombination. So one question, why are bacterial capsules medically important? So it's not easy to treat a bacterial infection, guys, with encapsulated bacteria in comparison with, or microorganism in general, in comparison to something that is non-encapsulated. Because when someone has an infection with an encapsulated bacteria, the immune system will be having more, well, it will be more compromised, it will have harder chances by itself to get rid of the microorganism, and also, in a way, the antibiotics, they don't penetrate as easily. what are the types of bacterial movement? Hmm? With the flagella, they do runs, tumbles, and then they can do also gliding, and then they can do twitching again. Michael Jackson. No, that's a moonwalk. Okay, now let's look into the cell wall. So we're going to compare in a greater detail the bacterial cell wall of gram-positive, gram-negative, acid-fast, archaea, and mycoplasma. Also, we're going to see different shapes of bacteria that are not the ones that we already saw. So we're going to see protoplast, L shapes, and how do they form. So what is the function of the cell wall in general? Well, it's to prevent osmotic lysis. Bacteria generally is found in an environment that is hostile, so they may have been in a place where there is a lot of water around and then very little solute, so they might have been encounter a hypotonic environment, and then the cells will tend to gain fluid, and that will make them to increase their volume and to potentially burst by the excess of volume. But because the cell wall is in a way more rigid and flexible, it will prevent this to happen unless you have a weak wall that can be done by weakening the wall with the presence of an enzyme like lysozyme or antibiotics. So in that case, yes, that weak point can make the cell to burst. Now, the cell wall also increases the chances that the bacteria produce infections. Not only the capsules, but also the cell wall. And the composition of the cell wall can help for differentiation of major groups of bacteria. Some of them are characterized based on that. So here is a TM showing you the different parts of the bacteria. So this thick material in here, it is the capsule, and underneath the capsule, it is the cell wall. So the cell wall, it is made out of a polymer that it is called peptidoglycan. Peptidoglycan is made out of subunits, disaccharides, that always are in this order, NAG with NAM. NAG stands for N-acetylglucosamine, NAM for N-acetylmuramic acid. That's like the name of the chemical structure, and muramic, it's coming from the Latin word muro. Muro means wall. So then you start linking these disaccharides, and then you form a chain, and these are going to be held together by polypeptides, and the rows are going to be held together by tetrapeptides. And all of this is assembled in the cell wall itself, and some of these components, these enzymes that help for this polymerization, can be the target of antibiotics, like for instance penicillin. So penicillin inhibits the formation of the peptide bridges that links the peptidoglycan rows, and that weakens the cell wall. And then these rows stack on top of each other, they form a lattice. So here is the image of these two, this monosaccharide that forms a disaccharide. So this is N-acetylglucosamine. So, I mean, this is sugar. And then it's the same as this N-acetylmuramic acid, except that it has all of this structure in here, while this one doesn't have it. And they always go like this, NAG, NAM, NAG, NAM, NAG, NAM, in that order. So here we have the rows of this peptidoglycan that are cross-bridged with these peptides, polypeptides, and you have these tetrapeptides that binds specifically N-acetylmuramic acid with the polypeptide that is from the previous row of this polymer. So that's peptidoglycan. Now, gram-positive bacteria has the peptidoglycan in its wall. So it can have like up to 30 layers. And within this cell wall, you not only have peptidoglycan, but also you have something that is characteristic of gram-positive bacteria, that is the presence of teichoic acids. Teichoic acids can be subdivided into lipoteichoic acids, which are found in the plasma membrane, and wall teichoic acids that are adhered into the peptidoglycan. Both of them are teichoic acids, but one adheres into the wall, and then you call it wall teichoic acid, and the other one, since adheres into the plasma membrane, if you remember, plasma membrane is made of lipids, you call it lipoteichoic acids. And as acid molecules, they carry a negative charge, and this helps to regulate the movement of cations. So peptidoglycan, the polysaccharides, and the teichoic acids makes bacteria to be easily identified. Like if someone is doing molecular studies and then they found in the sample that the sample has teichoic acids, or you can tell, well, this sample that is coming, and then it didn't know the identity before of that sample, it can tell, well, more than likely, this is the sample of gram-positive bacteria, because it has teichoic acid. So here is a magnification then of the external structures of these gram-positive. So you magnify, here you see the plasma membrane, and then on top you see the rows of peptidoglycan. And since you don't see another membrane, this has to be a gram-positive bacteria. And so here in this plasma membrane, you can see proteins embedded into it. And here in this peptidoglycan, you can see the adherence of this teichoic acid. And the one here on this side that adheres into these lipids is going to be called lipoteichoic acid. For gram-negative bacteria, we have thin peptidoglycan layer, 1 to 3 rows, and then this peptidoglycan, it is surrounded by an outer membrane. And the space between the outer membrane and the plasma membrane, which is where you have the peptidoglycan, it is called the periplasmic space. In this periplasmic space, the bacteria has enzymes that helps them to resist the action of antibiotics. A big clinical difference between gram-positive and gram-negatives is that gram-negatives are more resistant to antibiotics than gram-positive bacteria. And then the outer membrane, it has these lipids that we call lipopolysaccharide, LPS. And also this is a main characteristic of gram-negatives, just as teichoic acid is a characteristic of gram-positives. And of course, you have lipoproteins and phospholipids in the outer membrane. So oftentimes I compare like this space to the periplasmic space. This can be like the outer membrane. This can be the inner membrane. And this can be the peptidoglycan. So this can be like a gram-negative. And then if it was a gram-positive, we wouldn't have this countertop and this station here. So then if you have here enzymes that can destroy antibiotics and disinfectants, not only the antibiotics but also disinfectants, a gram-negative will be more likely to survive the attack of this versus a gram-positive. So what else does this bacteria in general, not only gram-negative, but any bacteria has to survive? Well, it has to survive not only attacks of antibiotics and disinfectants, but also phagocytes. We have in our body the natural killer cells, the NK cells. We also have the neutrophils, which are the first responders. So when someone has a bacterial infection or an infection in general, the first cells that increases in number in our blood is neutrophils. And then we have lymphocytes, B and T lymphocytes. So those are the phagocytes. Also in our blood, we have proteins that are called complement system that complements the natural immunity that we have, and then or are part of the innate immune system. And then of course, any bacteria that causes an infection and you treat that infection with antibiotics has to undercome that attack. Now, for LPS, it's made out of subunits. One that is the lipid A, and then the other one that is the O polysaccharide. So that O polysaccharide is actually the antigen that it is used to also classify these strains or variants of the species. So O157 then refers to this O polysaccharide in E. coli, part of the LPS. And then this lipid A, which is basically the part that adheres into the plasma membrane, it is what it causes fever in these bacterial infections with gram-negatives, because it resets the temperature center, the hypothalamus, so that we can have fever. And then the gram-negatives, they have porins or channels that allows the entry and exit of nutrients and wastes. So here is the bacterial cell wall of a gram-negative. So if you magnify here, you have a sandwich. So two membranes surrounding the cell wall. Cell wall is very thin, made out of 1 or 3 layers of peptidoglycan. The inner membrane is simply called plasma membrane. It has, just as any plasma membrane, channels, it has proteins, and then the outer membrane has this LPS, lipopolysaccharide. Lipid A attaches into the outer leaflet of the plasma membrane, and then you have a series of saccharides just connected to this lipid A that's called the core polysaccharides. And then at the tip of this LPS, the ending, you have the O polysaccharide. So this is the one that varies in different bacteria, and it can be used to identify these bacteria. So if you magnify then this part, you can see lipid A embedded in this outer leaflet of this plasma membrane, the core polysaccharide, and then the O polysaccharide at the end. So gram-negative bacteria releases this LPS, and particularly the endotoxin, the lipid A, during the death phase, or when the bacteria has been destroyed by an antibiotic. So something that, for instance, it is said to people that has treatment with antibiotics for a gram-negative bacteria is that expect to have fever as a reaction. So it's because you are basically releasing into the bloodstream this lipid A. The mechanism for why bacteria stain differently with a gram staining technique, well, it's because of the thickness of this peptidoglycan. So in gram-positive bacteria, alcohol in the second step dehydrates the peptidoglycan. That seals out the pores that could allow the exit of the crystal violet iodine crystals. And then, because you have seawall, this bacteria will remain purple in the next steps. While gram-negatives, because they have a very thin peptidoglycan, and you have the outer membrane, the alcohol is going to dissolve these lipids, it's going to create pores, and it's going to leave pores in the peptidoglycan, which after the washing step, after you have added the decolorizer alcohol, crystal iodine complexes are going to, crystal iodine complexes are going to leave the cell, leaving the cell colorless. And then you have to apply the secondary stain or the counter stain, safranin, in order to have these bacteria visible. Otherwise, you won't. It will be interesting to know, I guess, I mean, you can try it once you do the gram staining, to see what happens if you don't add the mordant. And then you continue with the rest of the steps, to see if the gram positives still remain purple. If you want, you can even reverse the order. You can add safranin first, and then crystal violet at the end, and see what happens. So this is then how gram positive look like: purple, gram negative, red. Now, some gram positives, not all, they produce some toxins that are called exotoxins. And these exotoxins, they have different effects in the cells, not bacterial cells, but in the cells that they are colonizing. And again, a difference between gram negatives and gram positives is that gram positives are susceptible, typically, if they're not resistant, to the action of penicillin. And they can be disrupted by the action of the enzyme lysozyme. Lysozyme is an enzyme that we naturally have in body fluids like our tears, our saliva, men's semen, also some respiratory secretions. Gram negatives, we all already know that they have four rings in their cell wall, the ones that have flagella, of course not everyone, and it produces these endotoxins, but also can produce exotoxins. And then it has low susceptibility to penicillin. Now, endotoxins then are part of the cell wall. And exotoxins are produced by gram-positive, mostly, internally, and they release it. Exo means outside, so that's the reason why they're called exotoxins. And the gram-positive bacteria that produces exotoxin produces these exotoxins during the growth phase of the bacteria. We will see those phases later on. But what it means then that there is a difference between when can you expect the presence of these toxins in the bloodstream of a person or an animal. So if someone has a recent infection with the gram-positive bacteria and the person starts showing right away the signs and symptoms, it's very likely, of course, that the infection is caused by a gram-positive bacteria. And the bacteria is actively secreting these exotoxins into the bloodstream. While the person will have a gram-negative infection, and the person, of course, the immune system starts responding within the first 3 days. And then as this immune system is destroying these gram-negative bacteria, the gram-negative bacteria die during the death phase. That's when you release the particles, the lipid A, then that's when you have the fever. Or if the person receives a shot of an antibiotic, of course, that is not penicillin that kills the gram-negative, in this case will be when you can expect the fever to happen. Okay, now some bacteria has atypical cell walls, like Nocardia species or Mycobacterium species. In the case of Mycobacterium, it has a thick wall made of mycolic acids. And these mycolic acids are found attached to the peptidoglycan. So this is a waxy lipid, and it helps for these bacteria to thrive more than even a gram-positive when it causes an infection. And then in this case, we use the acid-fast staining technique, whether it's Seal-Nielsen or the other type that I remember the name, but basically in that other one, you don't use the heat, while in the Seal-Nielsen, you use the heat. And when we already saw the details, like you use this primary dye, which is carbolphishine, and then you use the counter stain, which is methylene blue. And it's called this bacteria acid-fast because it resists the removal of this carbolphishine after you add this acid alcohol, which is basically hydrochloric acid with isopropanol or ethanol. So this is the view that you saw before. So this bacteria is acid-fast, while this other one are non-acid-fast, or even it can be cells that are stained with with methylene blue. Typically, the sample of someone who potentially has lung tuberculosis, it is sputum. So in the sputum, you not only expect to see bacteria. I mean, normally we have a bunch of bacteria in our oral cavity and part of the throat, the larynx. But if you see in some of these, I mean, these cells are large, so these are cells of the throat, and then the tiny ones will be bacteria, part of the normal flora. while these ones will be pathogens. Anytime that you see an acid-fast microorganism, it has to be a pathogen. It's not normal. Okay, so there's always an exception to the rule for having cell wall. Mycoplasma is the exception. Instead of having a cell wall, it has a plasma membrane, and it's still a bacteria. We characterize bacteria by the ribosomal RNA. So then, this bacteria has esterols in its plasma membrane, and these esterols, just like cholesterol in us, it will protect this bacteria against lysis. This bacteria produces an infection in the lungs that is called atypical pneumonia or walking pneumonia, because a person initially doesn't have like very severe signs and symptoms. And then archaea can be sometimes wall-less. And if it has a wall, it will have pseudomurine, which is basically the substitute for N-acetylmuramic acid. So in this case, it is NAM, the one that is lacking. It has pseudomurine. And these walls has the amino acids, which is unusual. Typically, proteins are made of L-amino acids. And some bacteria as well can have in their proteins the amino acids, not only archaea. Okay, so how can you weaken a cell wall? Well, by adding lysozyme, which is going to hydrolyze the bonds between glycans, like the NAM and NAG, and penicillin that breaks down the formation of these bridges. And when you do that, you end up with alternative forms of cell wall, like proplast, that it's a wall-less gram-positive cell, or an spheroplast, which is a wall-less gram-negative. You can end up with L forms, and it's not because it has an L shape. It is because they discovered it in Lister Institute in France. Any form of this, because it has a weak wall, it is more likely to be burst by osmotic lysis. So you can treat some bacterial cells with an antibiotic or with an enzyme or both, and then you put it in a fluid, in a liquid, and it would burst. So gram-negative, damage to gram-negatives, well, it happens less likely with penicillin, but you can use other agents that can penetrate the outer membrane. In this case, you modify these antibiotics, and that make it effective to inhibit the growth of gram-negative, like ampicillin, for instance. It can be more likely to damage this gram-negative. Okay, so this is the last slide for today. Why are drugs that target cell wall synthesis useful?
It disables bacteria's ability to prevent osmotic lysis.
Very good. Yeah, thank you. Why are mycoplasmas resistant to antibiotics that interfere with the wall synthesis?
They have the sterols that act as a different type of protection almost for the bacteria.
Yes. And as well, since they don't have a cell wall, they're not susceptible to something that can damage them. How do protoplasts differ from L forms?